Hello, 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 2700. Welcome back. It feels like we were just here, or it feels like forever has passed. It's this 2021 pandemic kind of a feeling where I think both feelings are the same. There's a co-instructor here today. This is Doug the dog that you may see in the background, or you may hear me talking to, or throwing a toy or distracting to. So, I hope you had a good break. I hope that you took some time and did some good things for your brain and your body, and that it was not school all of the time, or maybe even any of the time. We're going to jump right back in to the phylum where we left off in the Dun phylogeny, where we're talking about Platyhelminthes, the flatworms. The last lecture, it's been a while, right? So we had the week off, and then in the last week of lectures before the break, we had the guest lecture with entomopharms, and then you had your first lecture quiz. And so 2 weeks ago on Monday, we started talking about free-living flatworms. We talked about planaria and how if you cut them up into a million pieces, you win a Nobel Prize. That's not true, but a Nobel Prize winner did do that. And today we're going to be switching gears within the Platyhelminthes, and we're going to be talking about these amazing things, which include 3 distinct clades of parasitic flatworm that have some common and some common adaptations associated with being parasitic, but also have some unique adaptations as to whether or not, what kind of parasitic lifestyle, how many hosts they associate with, that kind of a thing. So you're going to see a lot of those kind of parasite life diagrams that when was the last time we would have seen them? Would have been probably when we were talking about toxoplasma. But there's a bunch of those today. And all those links and everything for that are, of course, posted in the chat at the end of the lecture. So I'm going to capture here all of your wonderful drawings, and I'm going to admire them for a second, and again figure out that there's a face when I did not see a face before. So holster your stylo. Here we go. That's it. Kids in our house rhymes a lot. So holster your stylo, and here we go. The very first thing is that I have been being made slightly hungry by a lot of these posts that I've been seeing that the course Twitter has been releasing and retweeting. You've been out making some amazing things with your cricket flour from entomopharms. So I look forward to seeing whether or not they're the cookies or the tacos, or Sherry was making this a little while ago, a butter chicken with cricket powder infused naan bread. I love it. I'm sure I would love it. And we tried some, Sarah Chorno did some, well, they look gorgeous anyhow. In our house, we've been making, we tried pancakes, we've been making crepes, banana chocolate chip muffins, and some no-knead bread. All of them were delicious. So good luck to you, and that's going to be a really fun lab session this week. Now, before we jump in to the parasitic Platyhelminthes, we've got, I have a survey for you to take, a mandatory survey, because there's an error in the course outline associated with the second lecture quiz. The first lecture quiz, which not everybody has written yet, and that's why the marks haven't been posted, but the second lecture quiz is on your course outline as occurring on Friday, March 12th. Because of a bunch of things, including the entomopharms guest lecture and the Demodex opportunities and things like that, I would prefer it if it occurred on the 19th, the next week. And so what I've done is make a survey on CourseLink for you to fill out. It opened a couple of seconds ago. That's for you to go and fill out. It's kind of a mandatory one. We're going to associate it with your capacity to do the pre-lab quiz for next week, just to make sure that everybody has their say. Why would you want to do this thing which I wish to do? One of them is we want to kick the can further along because we're not going to be through all of the things that I had hoped by the time we get to that next slice of lecture quiz. And by slicing out finals and midterms and talking about 3 kind of proportional lecture quizzes, that was a specific pedagogical design in this year of pandemia. My hope by putting it on the 19th is that I can keep them roughly proportional in size. And if not, the last one will be bigger because we won't be through all of the things, all of this vast morass of life. Why else would you want to do it? Well, if we do it on the 12th, there's going to be kind of lectures included in that quiz right up until the Wednesday before. And so, anyhow, there's the survey. For it to change, I need to get a nice 100% agreement. So here's my attempt. The quiz is open today until 4:30, and we will see what the results bring. Now, for something completely more interesting. You're done phylogeny, as we've been kind of leaving the radially or lacking symmetry early branching lineages with the fascinating who's first, the ctenophores or the poriferans, who's most the earliest branching lineage. We talked about the mystery of the purple sock with the xenocoelomorph, and then we jumped right in to the protostomes and the spiralians, and we've been talking about the platyhelminth flatworms. And you've seen this kind of looping GIF a couple of times. This is a parasitic one that we haven't got to yet because we've been talking about free-living flatworms. Today, we get into the gross, the nitty, and indeed the gritty. So a reminder, when we're talking about this particular tip, we've got examples that are marine, live in marine, terrestrial, freshwater environments, and parasitic ones. And that red kind of bar stamp, that is the one for representing parasitic life. That's where we're going to spend a lot of time today. A reminder what Brusca categorizes as the 7 kind of characteristics, or we say apomorphies, for the phylum of Platyhelminthes. They can be parasitic or free-living, and they're unsegmented. So they're worms, not in a true sense as the annelids, which we'll come to next week, but they are unsegmented worms. The cestodes, which look segmented, we'll talk about this today, are in fact strobelated. You're not segmented, you're strobelated. They're triplobasts, so we have a coelom, bilaterally symmetrical, dorsoventrally flattened. Doug, leave it. It's the point in the lecture where your 6-month-old puppy tries to eat a battery. Spiral cleavage, which is characteristic for most of the spiralians, but in fact not all of them. A complex, though incomplete gut, usually present, the gut absent in some parasitic forms like the cestodes that we'll come to today. They are cephalized with a centralized nervous system, and they have protonephridia, which we talked about last time we were together, as excretory and osmoregulatory structures. They are hermaphrodites with a relatively complicated reproductive system. And this is the kind of small clade colored to say all of, in the background, this basal kind of color of, I guess, yellowy khaki. Not bright yellow, but khaki, the platyhelminths. Then we've got free-living planarians, turbularians, in quotes, as a Tribbiani clade that is really not monophyletic, but represented and this simplified by one branch. Then we have the Neodermata, and that's the group we're going to be talking about today. These 3 parasitic lineages, the monogeneans, so we've done the free-living. We're going to talk about the monogeneans, the cestodes, which are tapeworms, or trematodes, which are flukes. And there's a rough difference. One of the reasons I like this and prefer this phylogeny to the one that's in your text is because it amplifies the fact that the endoparasitic clades, the cestodes and the tapeworms, are monophyletic, and the ectoparasites are, well, just this one lineage of the monogeneans. Working the door, everybody come on in. So there was a group back in ancient history, when I took this class, that we were taught the Cercomeromorpha, which was a group that was unified by the cestodes and the monogeneans. Get out. In the monogeneans, this was because of a particular modification on the posterior end where we see anchoring hooks associated with not being knocked off of your host. Now we know, because of molecular evidence generated since the last century when I took this class, that that is in fact not supported any longer. So the Cercomeromorpha, you can see that even here, they wouldn't be supported because they are, of course, that would not be a monophyletic group because it excludes the trematodes. And why would you want to exclude the trematodes? So we're going to start out today with a group that is exciting to me, the monogenean bladder flukes. Monogenea is They are an of the group, remember from that last clade, they're an ectoparasite. They have well-developed dorsoventral muscles. They tend to have aquatic vertebrate hosts. They have no intermediate hosts. So the who infects who is actually very, very host specific, very, very species specific. And there's not an intermediate host as we're going to see down the line, especially with the Digenean or two-host trematodes. There's no asexual stage, monogenea. And they have an oral sucker, the prohapter here at the anterior end, and the epistholapter here at the back end. This is a posterior attachment organ. And their larval stage is associated with being transmitted and moved around to ensure that it infects different hosts. So there's a look at the epistholapter and the prohapter, the buccal sucker. Here's what they actually look like and what you can see, that kind of foot. Some of you were for the hapter at the end, the oral sucker anteriorly and the hapter at the end. Some of you were coloring this in one of the lithographs that was in our welcome slide at the very start of the lecture. So let's take a look at the fact we talked about they have aquatic vertebrate hosts. These are important parasites, particularly in aquaculture. Those aren't, and there are a couple examples in your text that are developed slightly. But I'm going to talk about how they affect a vertebrate that's particularly near and dear to me, amphibians. In a previous life and in some parallel kind of research in my research program, I spent a lot of time caring very deeply about amphibians. And monogenean platyhelminth flatworms are particularly good parasites of amphibians. And the example we're going to dig out today has to do with this, a species of toad, a spadefoot toad. And I'm going to play this video. I'm going to tell you about it first, though, because if this ends up being cut out because of copyright infringements on the YouTube, I want to make sure that the asynchronously attending folks, hello, through space and time, he says hello, figure out what's going on here. This is basically a look at spadefoot toads, which are a toad that lives in a particularly ridiculous environment for an amphibian. It lives in the desert. And so when the rains come in the desert, life for that toad happens very fast. And then, of course, things for the parasite happen very fast. So let's take a look at this short video that I'll jump through that shows you some of the rains coming back to the plain, to the deserts in southwestern United States. Yes, I will, indeed.
Toads have to retreat once again below ground.
So,
the miracle.
A quick look there with the melodious tones of David Attenborough at a spadefoot toad adaptation, or adaptations to living in a desert environment. They're associated with speed. They have to respond very, very quickly. They need to come out of their little hibernacula underground where they make a little mucus ball and can live moist and safe within that. Respond to the water that returns in those torrential rains. Find mates. Males have to find females. Females have to find males. They have to choose. The females have to choose who they're going to breed with. And they have to successfully breed. Lots of amphibian fertilization is, of course, external and needs to happen in an aquatic environment. Now, the spadefoot toad that we're concerned with here is from the United States, Scaphus couchi. And this is a calendar year here. Take a look at the diagram in the top left-hand corner. So starting, we could start around now and say they're underground. And for the majority of that clock, maybe 80% of the year, they are estivating underground in their mucus little ball. They have a small window in the spring where the rains return and the males and females go out and call, and they're calling in these very ephemeral temporary water. And that is a critical time of year for Pseudodiplochorus americanus, which is this particularly gorgeous monogenean parasite. In the same window of time, preceding even the same window, as before the rains come, as different gonadotropin hormones change within the toads and encourage them to become sexually active leading into the rains, they also activate the trematode, the monogenean parasite. So the toads are estivating for 10 to 12 months a year. They have within them, if they're already infected, encapsulated onchomyricidia. And these are larval stages of the monogenean parasite. When the toads enter the water, the larvae are deposited by the adult, the previously from the last year infected adult frog. They pass out with the urine. They hatch within seconds. It's nearly instantaneous and are fully infective and can be infected immediately. They're looking often for tadpoles that are going to be, so as they kind of move around and respond to movement cues, they're looking for tadpoles. They want to latch on to the gills of those tadpoles and then slowly move through their body and then insist themselves, when the tadpole transforms into the adult, into the uterus or the bladder of the adult frog. So they invade the toad via the adult, if they're going to invade adults, it's via the nostrils. Then they go into the lungs, undergo a period of development, and finally then migrate to the bladder, passing through the stomach and the intestine. Now, take a look here. Particularly, you can kind of move your head really close to the screen. But when you've got this PDF after the lecture, take a look really closely at this. What life stage this monogenean is packed full of. So, transmission of this has a kind of a maximum of 7 hours to spread over 1 to 3 nights. I want to back up for a second because I realize as I'm talking, I gave you, in terms of the latching on to the gills, that's another monogenean for a different frog species that is in fact talked about in your textbook. So I apologize. This particular one infecting the spadefoot toad infects the adults through the nostrils. So just simplify that. And then there's other monogenean trematodes or monogenean parasites that infect other species of toads that have different transmission routes. So the spadefoot toad through the nose, spadefoot, nose. That transmission has kind of a window of 7 hours spread over 1 to 3 nights in which they can do it. So in some years, all of the toads become infected. And what you realize when you start thinking about the chronology of this is that this is an extreme synchronization of lifestyles and an extreme dependence of one species, the parasite, on the other, the host. Both species are, in fact, influenced by the host species' gonadotrophic hormones. So the host species is influenced as evolutionarily it was intended in terms of stimulating a sexual response. But what it does for the host gonadotrophic hormone does to the parasite is essentially gets it ready to begin its infective transmitting stage. One of the interesting elements about the biology here is that males that are parasitized by this monogenean have calls that are more attractive to females, which is kind of interesting if you think about it. Remember when we talked about, we spent a lot of time diving into toxoplasma and thinking about how parasites could manipulate behavior of hosts. And here again, when we spend a lecture talking about parasites, we're immediately going to be talking about several different examples where the host behavior seems to be manipulated by the parasite within. So in this case, females who are choosing the quality of their male based on different measures of their call, where maybe it's the amplitude, the loudness, or a particular burst duration. But the males that are parasitized have their call changed to make them more attractive to the female. And one of the interesting things about does this affect, so that's kind of a subtle tweaking of the biology, well, not so subtle tweaking of the biology of the host, but in fact there's some negative aspects, as you might imagine, that infected toads exhibit decreased fat storage after emergence from hibernation. And the fat storage is really important because if you're going to be estivating under the ground for 10 to 11 months of the year, the amount of fat that you can lay down is immediately correlated to how well you are going to survive. And I would even introduce into this, most of these studies have been done in the late 90s, early 2000s. And one of the things that we've seen since then is, of course, greater influence or greater impact, greater amplitude of anthropogenically forced climate change. And the climate crisis, one of the ways that we're experiencing it is, of course, in the redistribution and the lack of predictability with hitherto predictable water. And so toads like this that are ultimately dependent on that predictability of the water and their parasites may in fact be headed towards a doom cycle in the near future because of the water. If those that are infected, if all of them are infected in 1 year, and if those that are infected have decreased fat multiplies, and if we have several years in a row when the rains don't come for longer and longer periods of times, we have a greater likelihood that individual populations of these toads and their monogenean parasites will go locally extinct. I told you I was excited about this. I don't think I took a breath. But because of that, let's jump back over. Please tell me their feet look like little spades. That is, in fact, why they are called spadefoot toads. They have little tiny horny growths that they can dig and dig backwards and dig themselves underground. And yes, Sherry. Hello, Sherry. That survey should be available on the course link. There maybe on the line on the homepage there'll be a more button. Click more, and there'll be survey. What will we be able to see what we got wrong on the first lecture quiz? I haven't decided that yet. That's traditionally not done with midterms or finals, but what I will do is go through the questions and I'll talk about the ones where the majority or 50% or more, this is my intent anyhow, where questions where 50% or more of you got them incorrect, I'll break down those questions in particular. And yes, indeed, David Attenborough's older brother, Richard, was John Hammond in the Jurassic Park series. This is true. Okay. And now for something slightly different. So those are the monogeneans, the external. So those double haptors, the sucker, the buccal sucker, and the haptor, those are associated with the fact that generally they tend to be external and need to be moved off of, they want to resist being moved off of different organisms, their host organism. We're going to move inside the trematodes right now, the flatworms or the flukes. Now, the apomorphy for this group, that we have a larval epidermis that's a combination of ciliated cells and a syncytial neodermis. What the heck, Smith, is a syncytial neodermis? Well, as you kind of consult back in time, you remember we talked about the syncytial neodermis being a really good adaptation if you're a parasite living in an extremely nutrient-rich environment to absorbing that, because you have this syncytial layer of cells where the individual elements of the cells aren't broken apart, aren't clearly identified as modular forms, and so there's a sharing and increased absorptive capacity. These are those microtrichs that increase the surface area, and also the syncytium that makes those nutrients very easy to share and to transmit. Now, the trematodes have 2 or more hosts, so these can be super complicated and beautiful weavings of life cycles. We're going to talk about them. Mollusks are often the primary host, and they can be themselves, different species of trematodes, enormous health problems, cause health problems for people. So we're talking about this purple clade here, the flukes, endoparasites. So they have a mericidium larva, so not the oncomericidium larva that we saw within the monogeneans, the intermediate host, the mollusk, and then a definitive vertebrate host. And here's a look at, from your text in figure 10.3, at a particular example. So we still have an oral sucker, so an adaptation to extracting food from the host. A ventral sucker, occasionally like on the ventral side, associated with attachment. They can have a bulbous pharynx, then they have circular and longitudinal muscles. So again, we're talking about movement via a hydrostatic skeleton. So that not an external skeleton like a chitinous hard thing, not an internal skeleton as we see with our neighbors in the craniata, but a skeleton based on antagonistically laid muscles firing across a closed hydrostat or a coelom, or the inner body. So you can see the oral sucker up there. They have a genital pore and opening there. Now here's another look from another text that has, basically it pulls out different layers of muscle. I've simplified it in the bullets there to circular and longitudinal, but different, kind of slightly antagonistic, perhaps at right angles, perhaps at slightly less than right angles, and those have to do with whip-like motions or undulating motions. Now, when have we seen this before? Well, in fact, when we talked about free-living platyhelminth worms, I showed you a video of some of the marine free-living forms that have all sorts of capacity to move and to dance through the water, and just gorgeous, gorgeous motions. Now, next, we're going to jump into a life cycle of the Chinese liver fluke that is complicated. It's complicated. Would that it were so simple. It parasitizes up to 3 different species. You're going to talk more about these ones, I believe, in the lab. But basically, what we've got here is a snail that has within the snail 4 different stages where they infect as a miracidia, become a sporocyst, become a radia, and a cercaea. That free-living cercaea bursts out of the snail into the water, infects a fish, kind of insists to the fish, I will insist that I insist. And that these metacercaria in the flesh or the skin of the freshwater fish are consumed by the host. These then move back down into our digestive tract where the adults exist in our biliary duct and intestine, and then we scoot out the embryonated eggs passing out into the feces. So the first intermediate host is the snail. The second intermediate host in general, in particular with the Chinese liver fluke, this is the fish. It can also be another kind of an arthropod, another invertebrate or an arthropod. And then the definitive host where sexual reproduction occurs is, in this case, a vertebrate. In this case, it's us. And there is the definition of the definitive host, one where the parasite reaches maturity and reproduces sexually. And you knew that because toxoplasma. We jumped all over that earlier. So egg to miracidium, to sporocyst, to radia, to cercaria, to metacercaria, to adult. That's a lot. And remember, if we were going to present this kind of a cycle to you in the quiz, we wouldn't say, tell me what follows. I would never say, tell you what follows. What falls in between egg and sporocyst? Oh, it's the miracidium. I mean, because you can look that up, right? If I was going to ask you questions about this particular relationship, I would show you this kind of drawing of their life history. And then we'd ask questions about that. Okay, so don't feel stressed that you need to memorize these stages or these names. I'd like you to be familiar with them and about the dependency that results in, for instance, in terms of an increasing number of hosts and increasing vulnerability, yada, yada, yada. But we would never start from an empty diagram and I would say label all of these. That's, I don't think, a very interesting way to go about remembering and memorizing information. So, one of the ways that I like thinking about these trematodes is by thinking about Shaun of the Dead and zombies. And Shaun of the Dead is one of my favorite films in the world. Thank you, Edgar Wright and Simon Pegg and Nick Frost. Now, zombies, how does that have to do with trematodes? Well, we're going to talk about, instead of breaking out the Chinese liver fluke and how it affects humans, we're going to talk about another example, the lancet liver fluke, Dicrocelium dendrichitum. And this is an amazing one to me because of its infection of ants. And so the secondary intermediate host in this case is an arthropod. It's an insect. The vertebrate that it infects, it can be people, but traditionally it's cows. And some of you may even have come across this if you come from families or work that had to deal with cattle. I'm going to tell you a little bit. This is a kind of a quick life history diagram where we'll start down there again with the snail. The snail, the miracidia, the snail consumes the embryonated eggs. Within the snail, we see multiple changes from miracidia to sporocyst to cercaria. Cercaria break out of the snail. In this case, they are put into a slime ball that tends to be released by the snail as they're moving along. Those are then consumed by an ant. That is then consumed by a herbivore. How? Let's talk about that in a second. And then the sexual stages occur within that vertebrate. So Carney, when he talked about this in the American Midland Naturalist, which is one of the first places that this particular relationship was documented, talked about the fact that ants, and this is where we're talking about zombies, parasitized carpenter ants. So carpenter ants that have consumed these parasite-laden little mucus balls of the snail become sluggish. And they found slowly circular walking in riverbeds and streams for as long as 1 to 2 hours. Others were observed to remain motionless in the same vicinity for a similar period of time. They called this tetany, another word for your Scrabble dictionary. Tetany, a condition marked by intermittent muscular spasms. And he also used this phrase, in terms of its infection, which is a complex series of unlikely events. I feel like that might be a good t-shirt for this past year that we've been all through together. But this is what it's referring to here. So I just kind of do this, I'll jump through it again with little red circles. Eggs are shed from the infected ruminant, consumed by the snail. They hatch and move into the snail's digestive gland. Then sporocysts and cercariae are released when the snail produced an ant-appetizing slime ball. Remember that that's a critical stage. Why might a slime ball be appetizing to an ant? Well, we'll consider that. But the ant appetizing slime ball, so that then manipulates the ant to say, hey, consume me. And then we're going to pack your body full of these metacercariae, which is probably going to change your behavior and make you a little bit of a trematode-controlled ant zombie. And then when the temperature falls, the tetany, this intermittent spasm of the ant is caused by the trematode sitting inside of its brain, and then the ant remains it's migrated up and it sits on grass, which promotes its ingestion by the definitive host. This is an amazing. There's one of the videos that I'm going to show. I'm not going to show it together in class and stand here. I'm going to watch it with you. As a zombie myself, but I'll post it afterwards. There's a great video of this infective stage on YouTube that I'll post in the chat afterwards. And this is just a look at from a paper in Scientific Reports a couple of years ago of a case of this relationship with an ant called Formica acerva in Cypress Hills Provincial Park in Alberta, where they documented this infective cycle in cattle and other ruminants, on kind of indigenous or endemic ruminants living in Cypress Hills. And in this case, they used 3D histology of the ants to show there's actual physical contact between the parasite's non-encysted metacercariae, this larval stage, and the ant's subesophageal ganglion. And so you can think about these literally as kind of puppet masters playing the neural strings of the trematode of the ant intermediate host to make them migrate up the leaf and sit and lock on, and then sit there and basically wait for the cow to come along and eat them. I love that, and I wanted us to talk more about it. So what I'm going to do is say, I'm going to post that link for our Google sheet in the chat. And I'm going to do this while speaking, which is clever. And what I'd like you to do is to think about this, so that trematode eggs for this particular species of the lancet liver fluke can survive freezing up to -80. There's some other just biology. Snail infection prevalence increases with the age of the snail. So consider these two questions as you jump into your breakout groups in a second. What time of year would you expect most infected snails to appear? And why are the ants an ideal host? And so I will come over here and say, let's have a look at our breakout rooms. We'll create a bunch of them, move the Chesterfields around. And so about 5 minutes here, and then as always, send a representative of your group to the Google Sheet. The link is already in the chat, and tell me about what you've figured out, what you've thought about, reasons that you might expect what time of year that the most infected snails to appear, and why are ants an ideal host. Where's Doug?
What are they doing?
I don't know. They're in a breakout room.
Okay.
I'm still recording, but
Okay. He's in his crate or enclosure.
he was fine.
No.
In there? Yeah. Well, I'm sure he wasn't fine here. Yes. He's got to learn.
Yeah.
He fell off the couch. It was quite funny. Hi, Sherry.
Hey, Alex, how are you?
Oh, good. How are you?
Hanging in there. Trying to figure out the logistics for tomorrow.
The delicious logistics.
Yeah, people are posting some good stuff. So
Yeah. I saw some of the, what did I see? Cookies and was it tacos?
Yep.
And Sarah had, I forget what Sarah had, but I saw that she was out making stuff as well.
Yep. I talked to her earlier, and she was excited to participate. And a bunch of the students will have known her from last semester, so
Yeah.
she's going to come to one of the labs. I can't remember which one. I think tomorrow morning. So
Awesome.
yeah. And the dog's all excited. I made.
Yes, I didn't have to. I saw the video of the little.
I made dog pup cakes.
Oh, that's very good. Dougie would have been into that.
Yeah. Well, I made some flatbreads too, but Riggs was all excited. So.
Cool. All right.
He's not discriminating, so.
Yeah. The truth be told, neither is Dougie.
Yeah, exactly. But he's been walking around with the packs of cricket protein every time I leave him on the floor, so I figured I might as well make him something.
We haven't. Well, Doug. I'm sure Doug would do that too. But he's usually distracted by the other ephemera that the kids leave on the floor, like crayons. He ate. We found him at the tail end, I think, of the 15th crayon that he consumed yesterday.
Oh.
Yeah. So he had a very Crayola colored on our walk this morning as I was picking up his feces. It was a lot of colors.
Yeah. Well, at least they're coming through. That's a good sign.
Yeah, exactly. Yeah, and at least it wasn't the Lego.
Yeah. Oh yeah, we had our share of Lego.
Okay, I've closed the rooms, and so we're going to see people coming back. Okay, welcome back, everyone. I hope the rooms were well taken care of and that we cleaned them up in the break. And now let's take a look at some of... You tell me in the chat, I believe you should be able to see that I just moved this in front of the screen that I'm sharing, so you should be able to see the Google screen. I guess tell me if you can or if you can't. So what are some of your ideas? Why are ants an ideal host for this particular trematode? Small animal, huge numbers. You got it right off the bat. Huge numbers, share their food. Large numbers. Possibly more inclined to eat the slime ball. Yes, there's a real subtle bit of truth in that. When you're an ant or even most insects, one of the things that they're really, really prone to is loss of moisture. And so a slime ball is a particularly moist, juicy treat. And so they are more inclined to eat that slime ball. And then some jumping across, attracted to moisture, yes. So the moisture and live in large colonies. And so basically you're playing, if you're going to infect a particular species of beetle, you'd have kind of one of them in the field. But with ants, you're talking thousands, tens of thousands, in some species, maybe even hundreds of thousands across a large area where your ruminant may be grazing, where your next stage, your next obligatory host is living. So it's a great game. Great manipulation, great species or taxa to manipulate for the trematode because of the numbers. And then early spring, that's when the ants would be thawing, possibly mating season. What time of year? Warm weather, spring and summer. Snails undergoing hibernation. Okay, so lots of hypotheses about spring, and the eggs can survive up to -80, not that we see that in Alberta or in Ontario, but they can certainly survive that long period of time. But in fact, it's the fall. And if you think about it, that's because, so you talked about, your first answer has to do with probability. Why are ants an ideal host? Because there's more of them. It increases the probability of infection passing from the ant to the vertebrate host. And in fact, the answer is the fall, because the increased likelihood of the most infected snails appearing is once that stage of, in the sexual stage of the ruminant has occurred, where there's been feces that and grazing that has happened by the ruminant that is already infected or becoming infected. And so a greater period of cow poop. You want to have a whole summer's worth of cow poop out there that the snail is then in turn infected by. So it's in fact the fall. So that's cool. We can talk about that later if you are interested. And I put in here to vérifier le chat, mais I am already seeing the chat, so on va continuer. We're going to keep going. Now, just a couple of other, before we jump into the cestodes in the last few minutes, a couple of other trematode examples here where, and one of them we're not going to dive in and talk about it a lot, but because up to 200 million people a year are infected worldwide, the Schistosoma trematode is the second only to malaria as the most devastating parasitic disease in the world. There's some least irritating but less catastrophic trematodes as well. In fact, that we'll talk about in a second. But there's a couple of links there in the bottom afterwards where you can read about some of the parts of the world that have to deal with schistosomiasis as a daily part of their life. But unfortunately, we're not going to have time to break it out today. One last tiny nod to this particular group, the Apistodogastria, which is a group of the trematodes that is a particular parasite, mostly of fish or turtles. And I put these in here every year because I think I'd like to talk about parasites of turtles. And this is not the year to do it, but they have a large, complex ventral sucker, and they are out there. So when you think of turtles, think perhaps of this particular trematode that's infecting them. Now our last time is going to be within the Neodermata, within the tapeworms, the cestodes. This is a gorgeous bit, and this is perhaps where the most, maybe some of you have already been grossed out. More perhaps of you will be grossed out in the next 8 minutes. So these tapeworms have no gut. So the gut that we saw, the simple gut, some complex kind of branchings that we talked about in free living and the trematode and the monogeneans. The gut is absent here. And it's absent because they live essentially in such a food-rich environment with a syncytial epidermis that the syncytial epidermis is functioning as a gut throughout their entire life. So they're a parasite that's so adapted to living in this nutrient-rich environment that evolution has essentially modified their exterior epidermis into their gut because they live in the gut. Now, morphologically, now bear with me here, they're gorgeous. You might not believe that looking at this diagram here. There's parts of them that are gorgeous, maybe as our compromise here. The Scolex is the part that is gorgeous. This is the holdfast, or the bit of the body at the anterior end that is adapted towards holding on to some element, some microvilla within an intestine, large or small. And then as we move from anterior to posterior, we see the neck. The neck begins to strobilate. Remember we talked about the word strobilate when we talked about polypoid forms kind of sending out these little tiny pulsing medusoid ephyra that was within the cnidarians, within the jellyfish. This strobilation happens here because we see what looks like segmentation. You can see, I'm pointing, that's not helpful. Let's use the mouse. We need to figure out an invertebrate word for the mouse. These little tiny apparent segments are in fact we call proglottids. They're not true segments. They're not true metameric segments. What's a metameric segment? File that in your brain, because we're going to talk about that when we talk about the annelids, which is our first truly segmented form. But these are essentially broken off little segments, proglottids, of eggs. And so let's go back to the good looking part here. These are the Scolex. So the Scolex is the holdfast organs. All of these are different species of cestodes that have different holdfasts associated with the different interior environment, the different shape of the intestines of the species that they infect. They can be remarkably complicated, and so in fact, compared to some other groups, the taxonomy, our understanding of what species is which and who's infecting whom, with cestodes is not so bad, and that has to do with the intricate morphological adaptations that different maybe figments of your nightmares here are associated. These are different scolices for different species of cestodes that attach to different parts of the body. What might the nightmare include? Well, it could include shapes like this. This is, I believe, is a cestode of a shark, if I'm not mistaken. You can see if you're going to leave biological sciences and work in the film or television industry and work in particular genres associated with horror, there's probably room for imagination to kind of take, if a cestode looks like this, what happens then? Now, they don't all have to be looking like nightmares. This is, in fact, one that of a species of shark cestode that was named Zeusopex caraparis, because of its very apparent similarity to the cat in the hat. I love that. So these are other scolices of various cestodes figures from your text. Okay, last couple of minutes we've talked about, this is just a call out to another part of the lecture, or another part where we talked about, in general, we talked about platyhelminthes, but this is the neodermis of the cestode, the non-ciliated syncytial endodermis. So the entire body is essentially functioning as an inverted intestine with increases in surface area and cytoplasmic extensions that allow easy sharing. One particular life cycle with the canine, the tapeworm, the Diplydium caninum, where the intermediate host is a flea, but we've got dog feces that pass out proglottids, these kind of bags of eggs, from the tail and from the back end of the cestode. They are ingested by a larval stage of the flea, different stages, oncospheres and ciscursoides, that occur within the flea. The adult flea then infects via the blood the host, the canine, or in some cases the cat, and then the life cycle continues. And they can also then extend to us because we live very closely with our pets, as one of them was here earlier. So the proglottids, they strobilate. That name is actually, think about Pinus strobus. If you think about plants at all, strobus is the Greek for pine cone or cone. So it's not truly metameric segmentation, but you can see from the young proglottids, the youngest ones are the area that they're formed is right behind the Scolex at the neck. And then so we've got the oldest that are most distal at the tail end of the cestode. Now, what does the tail end of a cestode look like? It looks gorgeous, but a quick look at some of the oncospheres, these ciliated eggs that have 3 small pairs of hooks here and here. And what does the tail end of a cestode look like to send you off on your day in the last minute or two that we're together? You can consult the news all over the world and see lots of fine examples of the tail end of cestodes that emerge from different people as they consume sushi or other things, other uncooked things. The 5-foot long example that was prevalent in the news a couple of media that came wiggling out of a man's body after he ate sushi. Doctors remove 6-foot tapeworm through man's mouth. It's a slow news day, I suppose, when this happens. But you can take a look at some of these videos as they are moved in and around us as their definitive host. Now, that's all the time that we've got today. So I'm going to take a look. I'm going to stay in the chat for a section. for a period of time. If you've got some questions that that you want to have answered today, leave them for me there, and I'll try and answer as we move things up onto online. The PDF will be there in a second. So welcome back. I hope that your break was a restful one, and it's nice to be back and to share some time with you and to talk about these things that we're passionate about. For those of you who will have your labs tomorrow, and I'd love to show you Doug. He's in fact been moved into his quiet space. So I'll try. He'll be here on Wednesday. Doug needs to learn how to be patient around people that aren't paying 100% of attention to him. So Doug will return on Wednesday, when hopefully you will too. But I'm going to stop recording, and I'll see you on Wednesday. Take care of each other. Ta-ta.